It's my pleasure to introduce Rick Horwitz, who's the director of the Allen Institute for Cell Science, and he's going to start us off today.
Thank you very much, and thank you for coming. And this is going to be a great day. So welcome to the second Seattle Cell Science Symposium. Today's program, when I look through it, is simply awesome, and I really can't wait to get started, so I'm going to be fast. Our presentations today cover the gamut from of cell science in the Seattle area, and it even includes one person from British Columbia, sort of in that theme of our upcoming Cascadia secession. We also we not only have institutional diversity, we have topical one as well. We have speakers speaking on protein design, cell signaling, and computation, and from basic to translational science with some lightning talks at the end. I'd like to begin by introducing you to the goals of our institute. It'll be very brief, and the short cameos that you'll hear throughout the day describing our data, technologies, and things that you can get on the Allen Cell Explorer. So with that, I'll get started. And let me get to the... Yeah, okay, there we go. So the goal of the Allen Institute for Cell Science is to create a cellular state space, a stem cell state space. And in doing this, we're actually addressing three fundamental questions in cell science. One is, how do we define and characterize cell types and states? And that's a general word. They're phenotypes, which could be types and states which I think are not defined particularly well, but hopefully our work will help start to define that. How does gene expression in the environments in which cells reside lead to these cellular phenotypes? And the third one is, how do cells transition from state to state? We're starting with the pluripotent state, or the undifferentiated cell, and in our case, at least for right now, working toward the differentiated cardiomyocyte. In thinking of this last point, think of Virchow, the 19th century pathologist who said every cell derives from another cell. And what that means is that when we think about these transitions, we have to consider where the cell came from, i.e. the epigenetic marks, the post-translational modifications, and the organization of the cell itself, which may imbue some sort of hysteresis or memory within the cell. This is an enormously complicated problem, of course, due to the number of genes, about 11 to 12,000 expressed genes in a cell, and the number of proteins. So this seems very, very big, but if you consider the fact that structure dictates function in cells, which is a principle throughout biology, maybe studying the structure of the organelles, of which there are about 30 or so, 30 to 50 of them, might be a better place from which we can develop predictive models of cell states on the one hand, and also begin to understand mechanism on the other hand. So this leads us to sort of a multi-scale approach where we can start out by studying the single cell gene expression, the organization of the organelles in different cell states, and relate that to phenotype, and eventually working our way back to the complexes that comprise the organelles and so on, and meet the type of work everyone else does at the level of the protein and the gene. We're doing this in iPS cells. They're just fantastic cells for this kind of work because they're diploid, they have a stable genome, they proliferate, and you can induce them to differentiate into a number of kinds of cells. So they're just great because we can manipulate them as well as observe them. And we can grow them in organoids, which are closer to what their in vivo environments are like. So the goals of the Institute, the initial goals right now, broadly spoken, are to conjoin genomics with the positions, morphologies, and the dynamics of key structures and activities using endogenous GFP tagging and live cell 3D imaging, develop models for integrating these structures and for state transitions, both at the gene and the organelle level, and develop a platform for image visualization, interrogation, and output. We're organized in sort of a team structure, and these are very interactive teams, so the integration is key, and they span from the gene editing and the differentiation all the way out to the visualization and the modeling, and in the middle of it is high replicate imaging. Our institutional values, our institute values are critical. They're team science. This is all about working in teams. It's about open science. Everything we do is shared. And it's about community empowerment. The stuff we do, we share it, and we hope that it will help what you do as well. Our goal is not to steamroll you. Our goal is to actually empower you. So I'd like to give you a little look into what's going to come real fast. And Amanda is going to tell you about the genes that we've edited, which outline key structures in the cell, and some of the imaging we have seen with that. And she's going to say diploid cells with gene editing is a game changer in cell biology. Irina is going to tell you about mitosis. You're going to get a look into what a mitotic cell looks like for the first time, not just the spindle, but where all the organelles that she studied are. It's going to be pretty awesome. And Dave Williams is going to tell you how we're analyzing our data and how we can see things that you never thought you could see before. So stay tuned. You're in for a real surprise here. So with that, I think we're done, and I'd like to thank all of you for coming, and I'd like to also thank the organizing group shown here, and if they would stand up, Melissa and Caroline and Natalie, I don't know where they are, but please stand up. They did a terrific job. They're the one that chose you, or did not choose you, so be sure to talk to them, okay? And I'd like to thank our communications team, which pulled off this entire thing from the organization to the registration to what you're seeing here. So thank you very much.